So last session, one of the best talk I think, and I'm going to start. Professor Sowdhamini, as you know, you're very well known. I learned a lot when I was a student for listening to her lectures. So she will be talking about protein sequence structure function relationship. So I would like to invite Professor Sowdhamini to come here and deliver her talk.
Thank you. Thanks to the organizers for inviting me, especially Arijit. And over and above what Siddharth mentioned, what is important is the kind of people whom you have brought in to share the work. And I think really Arijit deserves a very big applause for bringing to the very nice set of people and topics to hear, and whole day I've been enjoying myself. And I also would like to add that I hope mine will be one of the good talks, because I've been hearing very good talks throughout. Okay, so this is not really the title of my talk today, so in case some organizers are panicking what's happening, because this is just the overall lab interest. Our lab is called as a computational approaches to protein science, or CAPS in short. And in fact, this year we celebrated the 25 years of the existence of our lab. Yeah, thank you. Thank you. It was all possible because of the students, and we also started it with a single sitting and single discussion. We decided to present the work that have been going on in our lab throughout in the form of virtual talks, and it spanned 18 weeks, one talk per week, and these are all recorded and kept in our lab webpage. It's called CAPS Talks, so you can go through them. They are just try to put them into different topics and so on. We had great fun. So just to say that our lab is interested in this broad theme of protein sequence structure function relationships, but in this theme I could have talked about how we are interested in intense and sensitive sort of sequence searches. We use SciBlast, and then we use it multiple times. In fact, every hit that we get as a homolog that we get as a hit, we would have given a chance to go in again as a SciBlast, and we call these as cascaded sequence searches. And we have been working on this for nearly 15 years. I could have talked about that, or I could have talked about a topic like, where different folds are coming in and seeing each other in these cascaded searches, or getting to use computational tools, say hidden Markov models of protein families, to then ask broader questions as to what are all the group of interacting proteins of a common function, such as our recent works on actin-associated proteins. That is, all those proteins that are known to be either directly or indirectly involved in interacting with actins, and how do these assemblies change in evolutionary time scale if we were to go across genomes such as human, mouse, and so on. So I could have presented this work in this conference, or I could have also talked about our recent interest to try and connect the genes that are common between cardiomyopathies as well as other diseases. And it was a pretty amazing sort of a learning exercise, because many genes, such as those involved in deafness, are also related in cardiac diseases, not only the diabetes, which one might expect. So these sort of themes are all coming under complex systems biology, or complex biocomputing, which could all fall very nicely under the theme that Arijit had weaved around. But what I chose to talk about today is some of our recent interest, a strong passion that the whole lab had developed for the past 10 to 12 years, which is to go after medicinal plants in terms of deciphering their genomes or transcriptomes. And into this, I'm going to just primarily talk about one such plant, which is called as the Sankapushpi, Clitoria ternacea, and the kind of genes, the fascinating genes that are useful in producing small molecules such as these by the plant, which have been exploited time immemorial and recorded in texts like Ayurveda. And the Sankapushpi, for instance, contain not only small molecules, secondary metabolites, but they also have proteins themselves that are present, that produced in high quantities, and these are called as cyclotides. As the name suggests, there is a cyclic structure to this. The first residue and the last residue of the small protein are peptide bonded. Plus, they are also held together by means of 3 covalent crosslinks. So we'll keep coming to this covalent crosslinks a little bit. And then what is known is that this conotoxins, for instance, they also are produced as proteins, larger length proteins. They have a gene behind them, and they have a pre-pro form, which means it gets proteolytically cleaved multiple times before it assumes its mature structure, which is the cyclotide domain we are talking about. It is also present not only in Sankapushpi, but many other plants belonging to many families, and it includes also coffee. And in fact, the term Robusta is provided to that family, that species of coffee, which has been producing these cyclotides at large amounts. And therefore, as you can imagine, from the plant point of view, it provides a pest resistance. But for the humans, it can give a lot of beneficiary activities. For instance, it was noticed in some of the texts that in the tribal populations, the flower of this plant is given as a tea to women who are just undergoing childbirth, because it eases the pain. So it reduces the pain sensation, but it also recorded in Ayurveda to have several neuromodulatory properties and being able to address some neurodegenerative diseases. Right? So in Sankapushpi, this particular cyclotide is produced in large amounts, and also this exact domain architecture varies from plant to plant. And you can see that the active and common domain is the cyclotide itself, but what are the coexisting domains may change depending on what type of plant we are looking at, but all of them have this sort of common structural theme, which is a cyclic moiety, and then there are 2 or 3 disulfide bonds that holds this domain together. And in terms of the disulfide itself, we could also ask how much differences are there between their sequences. The disulfide connectivity is highly conserved. Those are the cysteines shown as C in the amino acid sequence, but then the overall structure might differ a tiny bit, because in some of them you can imagine what is called as a Mobius, which is kind of a cyclic ring-like structure, but in some others whose crystal structures are available, the conformation is called as the bracelet structure, and it depends on what sort of peptide bond is present in this loop region, which contains a prolin, whether it is a cis conformer or a trans conformer, as far as the torsion angle of the peptide bond is concerned. However, it has overall remote resemblance to the trypsin inhibitors that are there in plants like cabbage and so on. So on the whole, they have some common sequences. The overall connectivity is the same, but the overall structure could be slightly different depending on the differences in the conformation of this loop region and so on. And in terms of asking how many such cyclotides are present in this plant, Sankapushpi, what we decided to do is to perform transcriptome sequencing. And the transcriptome sequence assembly is quite a complex biocomputing. And for this, ideally we should have had raw data, but since this is a non-model organism, we decided to start from the scratch, which is to go after each of these plant tissues, namely flower, leaf, pod, and stem, extract the RNA from them, and then submit it for sequencing. And as a result, we were left with 282 million reads, and each one of them are about 100 to 150 base pairs. And you might wonder, what are these reads and why are there so many of them? And if we ignore the long read sequencing techniques that are available these days, and especially for large plant genomes, what is typically done is after purification, whether you're looking at genome, namely the DNA, or the transcriptome, namely the purified RNA, typically they are just broken into bits. And then they are put together, and that is a process of assembly, which is a purely bioinformatics tool by which you try and do a jigsaw puzzle and get them all together to understand how the genome will look like. A little bit we have heard from Karthik before the tea, and that was in the context of bacterial genomes. But here the same is performed at 282 million reads, and then we use programs like Trinity, and the assembly tool is also pretty complex, as I said, because each of these fragments, how you put together, lots of decision making goes on, and there's something called a de-Bruijn graph by which you put in the fragments together to give rise to convincing bits of your genome or transcriptome, which you begin to call them as contigs or scaffolds depending on how large they are. The scaffolds are the larger bits, and if we had a very good assembly, then each of these scaffolds could correspond to chromosomes. So that's the ideal best scenario one would expect. But then what we had done was to use Trinity, and then you have parameters like N50 which will help us to know how good is the assembly, and then we were left with a large number of transcripts. Then we also do validations and make sure the essential genes are being seen after your assembly, and we found that 94% of the essential genes that we expect, like the dehydrogenases and so on, are indeed present, and we are able to pick them up from amongst the transcripts, which shows that the assembly is reasonably good. And then we quickly started looking for the cyclotides, and in doing so, we came across, we cared for also the coexisting domains, which means the N-terminal region, not only the cyclotide, but this N-terminal region, which is shown in lavender color here, and then the following albumin domain, which is shown in this cream color. And everywhere we were able to pick up on some examples. Initially, we did not get as many as the other plants were having, like coffee. So we decided to relax the minimum length that you expect for a gene. And then we were able to see quite a few cyclotides. And in doing so, we found that not only we are able to get enough number of cyclotides, as many as 71 cyclotides. So you can imagine that this is quite a huge number, right? Why should the plant be expressing 71 different disulfide-rich systems, even though it is good for its pest defense? Why it should do in a large amount? So to me, it is, in my mind, the analogy that comes is, imagine the pre-Google Pay era, the pre-Paytm era, or even the pre-card era. Our parents, for instance, were handling cash. And when they go out for a trip, they fill their purse with some 500 rupee notes, 100 rupee notes, 5 rupee notes, and so on. And depending on where they want to use the cash, the amounts will be more. So also here, the plant seems to be equipped to deal with a variety of pests. And it is just ready to express a large number of cyclotides. And as you can see, and much later, we will talk about it through the mass spectrometry lens, which is the sequence are slightly different, which means it should be able to do something slightly different to the insect. And in some might paralyze in some case, it might not let it eat at all in another, it might act as an inhibitor to their own proteases, and so on. So there are 71 of them, and out of them 26 are novel, in the sense that such cyclotide sequences have not been reported before. But I would like to really quickly go through another next 3 slides on the cyclotides, and then go on to the enzymes that help the plant to make the cyclotides. So let's go on to those. Here what we see is, given that you have transcriptome data, and then you have it on different tissues, such as flower, leaf, pod, stem, and we didn't include root here, is that you can put them in the order of how many million reads are getting aligned with your assembly. Then you go back to the raw reads and try to align the reads, and when you do that, you put them in the descending order such that you're looking at those which are highly upregulated in leaf are shown here, highly upregulated in pod is shown in this phylogeny, and this highly upregulated in stem would be shown here, and so on. So this immediately gives us an idea about what sort of genes, not necessarily only cyclotides, but in this case we have only used cyclotides, have the high transcript million reads, and we get to understand which tissue is holding highest amounts of what sort of cyclotides, and we know there are 70 of them. So definitely not all 70 of them will be equally expressed, and that is where the beauty of transcriptome sequencing is, because unlike a genome which will give you an idea what is the blueprint, here we will get to know how many of these genes are actually expressed in different tissues or environments, or depending on the process that they have gone through. And you can see this differential expression, which was very, very nice, but it also helped us to understand which tissue to go after. If you already knew, for instance, which of those 70 cyclotides are potent for a particular medicinal purpose, right? And those sort of in vitro assays are very much awaited. This is just a beginning. Okay, so this gives us an idea. But then we also did something which is because of our collaborator and coordinator, who's Professor Balram, and we went ahead and performed mass spectrometry analysis, and the mass spectrometry and the transcriptome in this case kind of support each other. The transcriptome data are strong because you have the nucleotide and then the transcript sequence information, but your assembly is not 100%. It's a draft assembly. So sometimes you have incomplete gene transcripts and so on, whereas a mass spectrometry can tell you how to fill in those gaps, and the same way for the others. So basically you do this for different tissues, and then you get the mass spectrometry spectrum, and we try to understand which are the major players in different tissues, what exact sequences are there. And you can see, although the cysteines are shown as conserved, shown in yellow color, quite a lot of variation is going on at the amino acids corresponding to the rest of the cyclotides, which means there is a lot of variety in the chemical groups and the properties that each one of them might confer. And finally, we also wanted to see how sequentially different they are, and how many of them are the Mobius type and the bracelet type, right? And to address this, we performed a phylogeny, and this is to show this mass spec work has been published. And then we went back and looked at the sequences and projected it as a phylogeny. And what you can see is the bracelet types are a large number, and they break into two different clusters depending on their amino acid variations, of course. And the third cluster is purely that of the Mobius. And the Mobius and the bracelet were the ones which has the cis-trans isomer difference that we had discussed earlier. Again, this sort of the data falling into neat three clusters is nice, but what exactly it means is something that we have to pursue. All right, so these are all to do with the cyclotide, but that is not the end of the story, because the plant is producing these cyclotides with the help of several enzymes. So we asked the question, can we go after the enzymes? And for this, we had to perform and standardize a computational pipeline, because the way we are searching for these genes or transcripts in a given transcriptome assembly is pretty complex, because if you're asking a question like an annotation that Karthik was mentioning, it may be relatively easy because you have a gene or a transcript at hand, and then you want to associate structure or function. But here we are asking the opposite question. We have a gene family that we are interested in, and into this entire transcriptome assembly, can we fish out all those that have the pattern or the sequence pattern of this family? And for this we rely on computational models, usually hidden Markov models, which are nothing but amino acid substitution data that you see have gathered over evolutionary time scale by looking at trusted homologs. If you're looking for a cytochrome, you pick up all the known cytochromes, build a hidden Markov model with that, and using those models go and search in your assembly. And this can be done both for transcriptome as well as, in the interest of time, I will skip this flowchart, but it is essentially to tell that you pick the members that you are very sure, create a hidden Markov model, and then search in the assembly of interest, put it back in the phylogeny, and see if it gets accepted with the other members or not. If they fall as outliers, maybe they are very different, or they could be false positives. All right. So with this in mind, we asked for, we searched for enzymes such as protein disulfide isomerase, highly involved in the formation of those cyclotides. And we see that there is not one or two, but as many as so many of these cyclic protein disulfide isomerase in this plant transcriptome. And later on we could see that there were in fact 15 of them that we accepted as true positives. And interestingly, if you cluster them with the other trusted homologs as mentioned in the pipeline, you will find that there are 3 Sankapushpi specific PDIs, protein disulfide isomerase. Apart from the fact that there are known members that neatly fall within the trusted homologs, there are newer members that have just emerged. This might indicate that these sort of PDIs are vitally important to make sure the cyclotides can be formed, because the disulfides have to be formed and so on. And they also looked after a few other enzymes, such as looking for asparagine endopeptidases, because these are special disulfide rich systems where the first residue and the last residue of the cyclotide have formed a peptide bond. And this is catalyzed by this enzyme, endopeptidases. And apparently in the commercial area, these sort of endopeptidases are very much in demand. So to our surprise, we found that there are as many as 7 such endopeptidases in the Sankapushpi transcriptome, and one of them behaved a little bit different, although it is a true positive, but it also has the properties to catalyze the opposite reaction, which is not a peptide bond formation but a ligase reaction. Again, in enzymes this is very common. The properties to form to catalyze a forward reaction and the backward reaction, you may need similar chemical groups. So all these again are waiting to be explored, but again we could see some Sankapushpi specific clusters. Right? And finally, we also looked at a few other enzymes which include the oxidoreductin and the cis-trans isomerases. And here again we could find quite a few trusted homologs that helped us to recognize as many as 70 different cis-trans isomerases. Whereas in the case of the ERO1, we found a single gene that had all the sequence properties when we started looking from the hidden Markov models and computer algorithms. Why are there so many of these cis-trans isomerases? Maybe these are recruited and vitally required for this plant to keep synthesizing variety of other molecules, maybe secondary metabolites, primary metabolites. We don't know where else these cis-trans isomerases are required, and whether the bracelet and the Mobius itself will be going through a structural transition while in the plant, we again don't know. We only have structures to show that there are 2 major conformers, and they also have some more diverse domain architectures. All this is very interesting, but what I want to do in the last few minutes is to take you through some fun modeling and molecular dynamics that we had done on the cyclotides themselves. And for this I would like to take you through a complete modeling exercise. This is not a homology modeling. This is not therefore a modular type modeling. This is not even alpha-fold modeling, but this is a 3-dimensional modeling of a different sort, which is specialized for proteins or polypeptides that we know are rich in disulfide bonds. And for this, the one and only condition or constraint that is used to propose a 3-dimensional model of a disulfide-rich peptide is the connectivity. What is the connectivity? It is the order in which the cysteines come together to form the covalent cross links. So if you have 6 cysteines like C1, C2, C3 and so on, the connectivity might say C1 is disulfide bonded to C4, C2 is disulfide bonded to C6, and C3 and C5 are disulfide bonded. So that forms the connectivity. You need that information crucially, and also it is useful to have the amino acid sequence. Otherwise you can just imagine all of them are alanine except the cysteines, and that's okay. But if we knew that where are the glycines and the prolines, it is better, because this is what this method called RAN-mod works. Random modeling. What is random about it? The conformation, the backbone conformation that is proposed to the peptide at any point of time is random, but allowed, stereochemically allowed. And depending on whether a residue is a glycine type or a proline type or a non-glycyl non-proline type, different allowed stereochemically allowed conformations will be randomly assigned, and that is where the randomness comes in. And it's also because for my PhD I was in Indian Institute of Science, right in the lab which comes from the school of G.N. Ramachandran, and the Ramachandran plot, the genesis of the Ramachandran plot had been in Madras University. So I think it's very appropriate that I talk about it, which is to assign 3 random numbers for each amino acid in this peptide sequence, which will then the programmed rand mod is going to convert them into phi, psi and omega values. The 3 random values will be converted that way, so that depending on whether it's a non-glycyl non-proline, which will be this particular box, this particular square, or whether it is a glycyl residue, which will be this particular square, or whether the sequence is a proline residue, which means it could be this particular square. And 80% of the time, the omega value, the 3rd random number, is assigned 180 degrees or plus or minus 20 degrees, so that you can expect anywhere between 160 to 200 degrees for omega, and 20% of the time the method, the algorithm rand mod will assign a cis peptide bond. So hope is that the bracelet sort of conformers are also picked up, and this then helps us to generate the backbone conformation. What happens next is a program called MODIP, which happens to be my PhD project, which is nothing but using disulfide stereochemical principles such as the side chain conformers and the distances and so on, to then ask the question, are the observed disulfide bonds strained or not strained? And this we had exploited to recognize sites where in a protein any 2 pairs, any 2 residues can accommodate a disulfide by means of engineering. So it may be a proline and alanine facing each other, but if you were to convert them to cysteine, can it effortlessly accept a disulfide bond there? And in doing so, which is called as MODIP, and geared by these sort of random backbone conformations that get generated, one could ask the question whether this disulfide rich polypeptide it is possible to generate a plausible 3-dimensional model or not. And therefore we can either accept or reject various conformations, and typically we do this like millions of conformations, and as and when these 3 disulfide bond connectivity information is accepted within the random conformation that has been assigned, it will throw out as a plausible 3-dimensional model. And this has been tested many times, so this is just to show you that given a disulfide bond connectivity, it will accept a random conformation, and then it searches and finds out whether it can accept a disulfide, and depending on the extent of side chain strain, they are graded and so on. But then it will give you models. Give you 3-dimensional models that does not depend on the presence of a homolog out there in the protein structural space. And we tested this, and then we applied it on the cyclotides, and we could see that several of the models that were proposed were indeed similar to the structures that we are getting from the transcriptome based assembly and subsequent 3-dimensional modeling, because we do have some structures of the cyclotides in the protein data bank. So this is the modeling story, but I won't take much time except to say that this Sankapushpi plant, and especially the disulfide rich cyclotides, have been very widely understood in the context of neurodegenerative diseases, not only childbirth, as I said before. So what happens is if you have amyloid aggregates or tau aggregates, the presence of these cyclotides in the system should actually disrupt the formation of these aggregates, or if the aggregates are present, it should start to remove it. So that is the basic hypothesis, and we did some behavioral assays. When we say we, it's really the student Neha Kalmanka who did all of these, and then she trained herself on handling C. elegans, and there was also already a transgenic C. elegans model which had huge amounts of amyloid deposits. And then she would ask the question, in the presence of the cyclotide extracts and different fractions which have high contents of the cyclotides, how are the worms behaving? And to our encouragement, we found that the extent of the aggregation is getting removed as seen by the phenotypic assay, which is how many of the paralyzed worms are able to move around better after the treatment. And this is the different concentrations, different fractions from different tissues of the Sankapushpi plant, and higher value means better, because we are now looking at the percentage worms which are not paralyzed. All right, and we can see that the pod, for instance, is doing very well, and so are the flowers, and so on. So then we finally ask the question, which is the imaging study shows that in the presence of these extracts, the extent of the aggregates are reducing, because the C. elegans advantage is the transparent one, so you can actually begin to see in front of our eyes through microscope, of course, that the extent of aggregates are reducing. And this has been published in this particular journal, and in fact as a cover picture, so we are very happy about it. But then we asked the question, can we validate this using molecular dynamics? And for this we picked up different amyloid aggregates which has a 2-fold symmetry, 3-fold symmetry, etc., 4-fold symmetry, and so on. And then every time we would impose the cyclotide, which is shown in the cyan color, and then performed molecular dynamics with and without the cyclotides. And we were closely following the interaction region where the cyclotide is shown in blue and the tau aggregate is shown in green. And then we wanted to not only see whether the interaction is being strong throughout the simulation, but what is happening to the green guys. The green guys are the aggregates, right, the amyloid aggregates. And when we do this, we can visibly see through the simulations over time, simulation time, that the extent of the beta strands that you neatly see are becoming disarrayed. The extent of the beta strands are reducing, the extent of the interstrand hydrogen bondings are reducing, and so the entire sheet is just melting away. And this seems to be conceptually easy to appreciate, at least for me, because the cyclotide is a very strong protein, because it is cyclized, it has 3 disulfide bonds. So the presence of this particular cyclotide is enough to melt away the tau aggregates. And this is a kind of a molecular dynamics exercise that helped us to pursue this analysis. So with that, I will really stop and acknowledge the people who have worked with me in this project. This is Neha Kalmankar, the student who carried out the entire analysis. And this is Radhika Venkatesan, who is a protein insect interaction expert, now moved to IISER Calcutta. And the entire project was coordinated and driven by Professor Balram, who also happens to be my PhD guide. Thank you. With this, I'll take any questions from my
questions?
So when we go back to ancient texts about therapeutic uses of medicinal plants, is it mentioned anywhere, or are there any signatures to say that the certain medicinal plant is rich with cyclotides, because then one doesn't know it is due to cyclotides or phytochemicals?
Yes, the exact chemicals that are behind it are mentioned less, but they would say rasayana, rasa. So meaning the ras is the chemical, but they may not know, because those days they don't have mass spectrometry. They only go by the phenotypic behavior. So in fact, even for the tulsi, which I did not talk about, there have been ancient Susruta texts which said Krishna Tulya Tulasi. So meaning the Krishna Tulasi and Rama Tulasi have equal amounts of the secondary metabolites. That's what they infer. But it is not because they know what chemicals are there. Definitely they would not have known that there is a cyclotide and what sort of cyclotides. But they roughly know that these chemicals are responsible. And they're purely driven by phenotypic and behavioral aspects.
So my question is there any indirect way to infer that a certain medicinal plant is rich in cyclotides?
Yes, this is how it is being done over time, because of late, if you look in the literature, not only for Sankapushpi, but for many other plants, that have been reports of cyclotides, and that's how we get to know, because of these biophysical equipments and techniques, and that provides the scientific evidence for the basis, as a basis for the ancient texts.
Thank you, ma'am. Regarding the Sankapushpi properties for the health benefits of whatever, you mentioned that there are 70 cyclotides. If I'm not from the same subject, my terms are maybe wrong. If whether they express according to the temperature or region, or like for example in Chennai they have a they might produce different health benefits, or in some other place do they produce different health benefits? Is it something like that?
Yeah, it's a very important question, especially if we are dealing with transcriptome data, because definitely it's very, very sensitive depending on the environment, the day, of whether it is morning or evening, or where the geographic, the soil texture, its pH and so on. For instance, there is also a Sankapushpi, the one which will produce white flowers. They are known to be more potent according to the Ayurvedic text, but we have not explored them. We are now looking at subtypes, but even in the same plant, depending on whether you grow it in Chennai or in Bangalore, the extent of this cyclotides could be varying. So that's why I think you also brought in an important point that I wanted to mention, which is whatever we have shown here and shared the data is really just the tip of the iceberg. So we can go back and do a number of other experiments to follow how the environment and geography affects the transcripts, and also to perform meta-analysis of the current data, because we have seen from stem, from root and flowers the cyclotides are different. This is only just looking at 2 types of parameters, but even with the current data we can do some very nice complex calculations and analysis.
Madam, it is a very nice talk. The same thing, I am also not from the subject. Still, whatever I understood, based on that, I just want to ask one question. That do you think that with this climate change, because I mostly work on the climate change, this kind of cyclotides has any, climate change has any effects on this cyclotide? Or they cannot change.
Yeah, you mean the global warming?
Global.
Definitely it is impacting the plants. We feel very happy when we see flowering plants out of the seasons. Okay, as long as we see flowers, we are happy. But apparently in the other areas like Atlantic and all, suddenly out of the climates, some plants are blooming. And then on the other hand, in Bangalore, on the news it came up that certain plants which used to bloom very well are drying up. So nobody currently understands why, and definitely the global warming and climate changes are impacting, and plants seem to be some of the sensitive ones which can tell us very early on. Definitely it is changing.
Okay, thank you.
Hi. So my question is that is it possible to do some kind of a negative control where plants that are not known to have a medicinal effect and test, are there these cyclopeptides in such plants that are known not to have a effect?
Again, it's a very important question that you have brought up, because I have been reading books, papers, reviews out of my own interest on medicinal plants and Ayurvedic texts. And to my knowledge, I have never come across a plant which did not offer medicinal value to humans. And usually it's like one plant, many diseases. Many diseases, happier the humans are. But then to even recognize one plant that is not going to be useful, it's very hard to imagine. Even the grass, it confers UV radiation protection. Many of the plants have antipyretic, antiviral sort of properties. Yeah, so it's going to be very hard to find that control. I would like to see it.
Hi ma'am, I would like to know if this was the first time the Sankapushpi was sequenced.
Not really, because there is one very strong group led by Craig in Australia, Queensland, Australia, who have been looking at cyclotides in this particular plant itself for over a long time. Surprisingly, this never bothered Professor Balram. He was just happy to find another group who was working on it, but I mildly pointed out that they also have a database, and it's fine with us. Some other group is already working on Sankapushpi.
But the strain would be,
the strain would be different. Like the other person said, the fact that we are collecting the samples at 2 different continents means that we are not really competing, and in fact we are augmenting data. And they had not looked at the associated enzymes like the protein disulfide isomerases. They were not able to find those many novel cyclotides.
So it's a very novel data from India, right?
Yeah, from India.
That's
we can say
lot can be explored with the data itself,
absolutely.
not just cyclotides, but because you have the entire transcriptome, one can look at all the other proteins that might also be useful.
Absolutely.
And have you looked at it from the point of the function for the plant? Like you said that it is helpful for the pest resistance.
Yes,
I'm not sure
true.
if I got it.
Yeah, true. The Radhika Venkatesan, who's a plant insect expert, had imposed the plant in presence of some well-known pests that are laboratory pests, and she could find that none of the pests come in front or near the plant ever. In fact, just so that the growth is good, we kept the plant out of the greenhouse and we used to grow it in the natural condition. And around the plant there are absolutely no pests.
Hi, it was a nice talk. So I have one question. Since you came up with the fact that a single plant might have like multiple activity for like antipyretic and things like that, so I'm curious to know if you tested this for antimicrobial resistance, because since it has disulfide linkages and there are some cyclopeptides that have disulfide bonds bridged, and then they are known to for antimicrobial resistance.
Yeah, I must say that different agricultural centers, agricultural science centers go after medicinal plants which are endemic for their own area. Like that ethno-medicinal plants in Western Ghats, in Arunachal Pradesh. So such groups have already been looking at basic assays such as antibacterial, antiviral. So for Sankapushpi also it must have been documented. So we decided to go for the other higher end questions, such as going after the cyclotides using transcriptome and mass spectrometry. But antibacterial assays, they just get done quite soon by many groups. Yeah.
Ma'am, yeah. So, I mean, discussing with some of my friends, they told me that Ayurvedic practitioners themselves do not subscribe to the fact that only one particular chemical in, the, I don't know, the oil or whatever the patient is administered with works. I mean, you need to give the entire thing. And so the question is, are there not scientific methods to test the whole, concoct or whatever, rather than testing the individual?
Yeah, that sort of understanding is slowly emerging. Because when I started in 2013, some of my friends and colleagues used to tell, all this is well, scientific evidence is well, but show me that one single molecule that is working for your plant. And now I can tell that it is never going to be like that, because we are asking the wrong questions, so it seems to me, because I did not talk about moringa, where we had gone after 3 major constituents, chlorogenic acid, benzylamine and quercetin, and we found that even if you do the same assay, with pure benzylamine, we will get good results. But after an amount, it becomes toxic when you do the toxicity assay. So perhaps it was never a good idea to take that one single molecule and inject it to humans. And the way the plants are dealing with in the midst of other molecules, not only make sure that the concentration of this compound is less, but also the multitude of other compounds are simultaneously doing other things. Like some may be antipyretic, some may be anti-inflammatory. So I think we have to start to respect that extraction that's coming from the plant to cure that disease or multiple diseases.
Okay, let's thank the speaker for a nice talk.